In this video, we will discuss about this FASL ligand expressed by endogenous and CAR-modified T cells mainly. So why the researchers focus on this FASL ligands? Because it has a role towards this cancer diagnosis that we will discuss in our subsequent videos. First, they check how they come up to this point is that when they check the cancer related data that is present in the cancer atlas, they are able to see that around 73% of the cancers has this FASL gene overexpressed. So what it is, but those data are based on bulk RNA sequence, and because of that they are not able to categorize from where this FASL ligands is coming from. So in order to check that, they did the single cells analysis of the 34 patients and 37 patients and the 4 healthy patients, 37 patients and the 4 healthy donors. And when they are able to do the UMAP, they are able to see around 18 different cell types based on the cell markers, and they are able to see 9 clusters are formed in the UMAP. When they check the FAS expressions, they are able to see that FAS expressions are highly expressed in this case of the CD4 plus T cells and the CD8 plus T cells, these regions that can be seen here in the FAS. And when they check the gene expressions, they are mainly expressed in the CD8 plus T cells. So this indicates that these T cells are expressing high FASL ligand, that the endogenously T cells has a more capacity to express this FASL ligand. Then they also checked it in the different categories that is been shown here, and there one observation they are able to see that these malignant and stromal cells have minimum to negligible expressions of this FASL genes that can be seen here, and whereas the T cells has the highest expressions, this non-lymphocytes also has the minimum expressions. So this lymphocytes subsets has the maximum expressions, that is T cells, NK cells. And thereafter they also check the in vivo results using this RNA fish studies, where they scale, and they are able to see that FASL ligands are present along with the these CAR expressions and CD3 expressions that can be seen in this panel D figure. When they quantify these results, they can see that the FASL gene expressions are significantly higher in the case where patients are treated with CAR T cells. So the patients for which this data is analyzed is being treated with the CAR T cells, and they are comparing the FASLG expressions in the CAR T cells and in the normal T cells. So when they expressed the CAR T cells, they are able to see more FASLG expressions compared to the case of this endogenous T cells, and this is statistically significant. This rise is statistically significant. And when they check it about this FAGL positive cells, and they are able to see that it is also high in the case of FAGL percentage positive cells. They are able to see that in the case where patients are treated with this CAR T cells has the high FASL ligands compared to the case of this endogenous T cells. So this in all together indicates that this FASL ligand is being predominantly expressed by the lymphocyte subsets, and when the CAR T has more expressions of this, when the patients are treated with the CAR T cells, then CAR T cells in those patients will have more expressions of this FASL ligand, FASL gene, compared to the case of endogenous T cells. So this is what they have found out in this results that are presented in this figure. If you need to understand more about their experiments in detail and their bioinformatic analysis, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.